known Candida for now many years, almost 15 or so, and where we were at Shire previously together. And so it was really fascinating to hear that, because I'm going to add and complement to, again, some of the disconnects in terms of the disciplines. I think that's the theme. How if the different disciplines in product development are not really thinking about the other aspect, then there's lots of money lost, unfortunately, and time lost. And those would be some of my examples. But before I go there, so for how many people know what CMC is, actually? 1, 2. All right. Oh, and I know you. Hi. Okay, good. 4. Okay, so CMC, the word stands for chemistry, manufacturing, and control. And what it stands for is everything you need to do to make the physical product. To actually, so here, in a one snapshot, I wanted to show, in the business of, at least in the biotech, but in any pharma, you have. Let me see if I can turn this on. Okay. So we talked about the clinical, preclinical development. We talked about, I guess, marketing and business, perhaps the BD part of it. At the end, the regulatory has to accept it, so you have a product. And then discovery, finds some sort of a target. But this whole piece here is what the CMC does, which is develop a process. And I'm focusing on biotechnology product. It doesn't matter. The chemistry part, if it's a small molecule, you're actually figuring out how to synthesize the molecule, and then it has to be manufactured. There's your quality control and quality assurance, and then the interaction with others. So this is the part that, and some of the mistakes that I want to be, or I would call it actually missteps that I would want to talk about today. I guess for those who know what CMC is, any ideas, or have you seen any missteps that maybe you want to bring up? Right, I use my example. Can I bring up one example?
Not that I'm allowed to talk about.
Oh, okay. All right. Well, let's see if we find common themes here. I have hundreds of examples. So, and I don't need to mention any company or any name of any product, because the themes are very similar, actually. So, I'm going to start with the first example, where it's this is a transition from what I mean by research, from research into development, the big D. So now you think you have a good candidate and you want to go into development. So in this case, this was research had identified a wonderful cytokine anti-cancer agent. We had different variants of that, different amino acid sequence and variants of that we had to be selected. But it turned out that it was a little difficult. There was some inconsistency in the animal model efficacy results. Okay, yes. There was some inconsistency in the animal model results, and it was difficult to find out, so which candidate is really good. So the D part, I was in the D side, I was actually physically involved in these, became involved, and we started collaborating with research, and it was actually really my curiosity. This is a very, very old program, but that I'm referring to. But started talking to the people who were doing the study, and I found out that they purified the protein, kept it refrigerated. It was actually put in PBS, phosphate buffer saline. That's so common. It's the most, most physiologically common buffer. It's at neutral pH, so it's physiologically very good. And the way they were dosing the animals is that they had these little. It's called minipub. It's like a little reservoir that goes underneath the skin, and it delivers very gradually over 3 weeks in the mice. These were mice, cancer model. And so the drug is sitting at 37 degrees for 3 weeks in this buffer. And so I started looking at pH dependence and temperature dependence of stability, and it turns out that the protein is aggregating really badly. It does not like PBS. And the best pH is actually to go lower, to be in the acidic form, to stay stable. So we changed this, and of course, research had never heard of the word formulation, By the way, I didn't before I came to industry either. I didn't know what that word means. But it was really important to know about the stability. How do you keep that protein that you think is going to be the wonder drug, keep it stable, so you can actually do the study. So anyway, we changed the formulation. The animal studies became more consistent. We actually saw not only good results, but also were able to select which clinical candidate to advance with. And that was with doing more structure activity and structure stability studies. And some of the candidates, for example, they had a free cysteine. That means it's more reactive. It's never going to be stabilized. So it was not a really good candidate. There are better ones. So this program continued. It was very exciting. We made it to to development, and Yeah, it went to basically IND enabling stage, so moved from research into development. So then we went into tox studies, and this is a monkey tox. Typically, you have to do some sort of toxicology in animals, usually rodents and monkey if it's a protein. And it showed severe nephrotoxicity, and the program dropped. It was very promising, but the program dropped. Now, it so happened that during the collaboration between R&D, between research and development, we had done some biophysical studies looking at the interaction of this particular cytokine with the receptor, with the cell receptor. And it turns out that the protein itself makes a trimer. Three of them come together, and it actually trimerizes the receptor, and that's how it becomes active. It triggers intercellular signals. And this was for the human protein. But it turned out that the monkey protein, in the monkey, it doesn't actually trimerize. It actually dimerizes the receptor. And then the research did some studies and showed that actually that triggers a different pathway, and it calls cellular toxicity. So here we're dosing a human protein in a monkey, and it's showing toxicity. But it turned out that there was one amino acid difference between the monkey and man, and that amino acid difference was in an apex which brought the trimers together. That was missing. The monkey protein didn't have it. And so, and by the way, all of this work was done underground because the program was dead. So the researcher who believed in this product, got some money. We made the monkey protein, put it in the animals, redid a small version of the tox study, and it was clean. And the program actually, revived. It came back. He went to the CEO and said, "I want this protein to move," and it did. And it was really that collaboration between, around the biophysical characterization, looking at stability, looking at structure and interaction with the target, that helped, which came from the development folks that think that way, came to research, and we actually were able to, revive a program. That's just one example. The other example was a little less fortunate. So here was, and what I would call it a sort of uninformed decision making, and having a go decision when it was really, or misinformed, I should say. But so here's the case. It was an enzyme. It showed great efficacy in animal models. So great program went into clinical development. The process development group, which is where I was in, started, developing a way of manufacturing it. And when we made the molecule in PD, process development, it did not show, send it back to research, and it didn't show efficacy. And you can imagine, and it also has solubility issues and whatnot. So you can imagine, at that point it's fingerprinting, PD does not know how to make proteins, and it was really bad. But anyway, so we went back and looked at, so how do we come to this, what had to be, actually there was an investigational team, which actually took about 6 months because there was a lot of other issues happening. But what happened is that the efficacy studies were done with the mouse protein, with the mouse enzyme. And obviously we were developing a human enzyme, right? We're going to inject humans, not mice. And it turned out that these two have very different properties. The glycosylation is different, the charge profile is different, and between the mouse and the human protein. And more importantly, the human protein didn't have any activity on the mouse cell. So it was not showing any activity. It was very difficult. And an interesting thing was that at the same time, so we had done market assessment. This was going to be, it's an orphan drug, but it was going to be marketable. And the company was successful in having other marketed orphan drugs. But the assessment was that, we should be able to, develop this and, make so much money. It turned out that as we started thinking through how we're going to actually even develop this, it was going to be almost impossible to find patients. So on the side, this is happening. So on the CMC side, there's a problem. On the other side, it's like, how are we going to even find enough patients to do a clinical program? And of course, the program died. But by then, we spent about $1.5 million, to just find out that this was not a way to even start the program. Again, it's kind of sad when you think about it. I could have used that money, but anyway. So, bad decisions when you really didn't look at, I guess nobody thought that this would be a problem, but bad decisions. So. Another I'm going to show you really bad examples, but it doesn't mean that everything is bad. But things that really surprised and had major impact. This one is actually even more impactful. And so this is another company that I actually happened to consult with. I came in right in the middle of the problem. So they had a wonderful program. It was in phase 3, ready to submit the license application, blood factor. But so when you make proteins biotech-derived proteins are made in cells. Those cells are called host cells. For example, it could be a CHO cell, which is a Chinese hamster ovary cells, or E. coli, or whatever cells that you produce it in. So these cells have thousands of proteins. You can imagine, as an intercellular proteins, there are thousands of proteins that you have to separate from your key protein of the product that you have to purify, right? Now, typically, not every host cell protein you can get rid of. So there are thousands of them, of which you probably end up with maybe a handful at the end in the product, but at ppm levels, so nanogram per ml levels, very low levels. So host cell protein is something that, as an impurity, we always have to check for. Anyway, the patients had shown antibodies to the host cell proteins. Now, it was really interesting. How did they even find out? So apparently, not in every indication, people look for anti-drug antibody. That you have to always do. Does your protein generate antibodies in patients? Because it could be anaphylactic or actually decrease efficacy. In this case, the agency had, they require for all blood products to also look for other antibodies to your host cell if you're coming from biologic products. So they had seen that there were no clinical impact. There was no anaphylactic. There was no sign of any clinical efficacy impact. However, both EMA and FDA actually put a halt on them. So here they filed to get licensure, beautiful phase 3 data, and they put a halt and said, go and figure out what's going on. You have to do an investigation. You have to find the root cause, the source of that. So we went back to history and the history of the program. What's going on here? And it was a kind of, unfortunately, really silly problem. So so that's where I became involved. So in phase 1 development, I found out that the assay that you use to quantify how much of this host cell protein you have, it was a generic assay, off the shelf that they had used. They didn't really characterize it very well, so it was not sensitive. And even orthogonal methods, there are other methods of also looking for this impurity, which is like running a gel and staining it. It was not stained with something that is sensitive enough, so they were about 1,000-fold off. And so there was enough, instead of having nanogram of these different, it's not one protein, it's a whole mixture of proteins. There was microgram. It still is very low, but that microgram was enough to create antibodies in patients because they're dosed. It was a chronic dose, so you're injecting it more than once. And so, of course, the company had to invest to go back, develop a new assay, develop new process, but by that time, I was actually really sad. The company went bankrupt. They just didn't have money enough to carry through. So program died, company went bankrupt. They couldn't actually pay myself. They couldn't pay my consulting fee. It was really sad. They told me, hey, you can go and, argue with the court. It's like, forget it, And it was all around basically having the wrong, poor analytics, to even know what to develop. It's really sad, actually. It shocked the hell out of me. But it turned out that a company of a few years later, actually a couple of years later, actually bought that program, went back, and it took them 2 years to, get the agency to agree to and whatever, and they went and they actually got approval. But, it took a long, long time. Anyway, so back to the question, what are some of the most common missteps? So now the question to you, which one of these do you think is the answer? So, you want to guess? No secrets.
If I had to guess, it'd probably be number 2.
Okay, it's actually all of them. So the case is, I've seen disconnect between research and development, right? The wrong molecule, whatever. Definitely, people just do. Formulation is like nobody knows about it. By the way, talking about who's your customer, I found out that in the teams that we would sit in, product development teams in companies, people really don't know about CMC. Right? Most of the development teams are driven by the clinical program. How are we going to do it? How are we going to get through the clinical program? And so I had to actually sit down with my clinical, colleagues and explain what do we have to do in the CMC? Because for them, it was like the person you're sending to the team. Why can't they make a decision right there? And I told them this is not one person who's designing clinical trial. It's actually half the company who's trying to, from QA to manufacturing to analytical to formulation, that you have to bring all of that together to make a decision. It's really complex, actually. So our customers were actually the teams and the colleagues on the teams to really actually educate them how we can make decisions and why we need information from them. Another example was, so I was in the pharmaceutical group, and the clinical folks, once they want to go and administer a drug to the patients, I needed from them what IV bag are you going to use, and usually that was the very last thing. And it's like, oh, we're ready to to go to patients. It's like, well, I cannot show you that you can actually put this drug in that IV bag until you tell me. It takes me about 3 months. So think about it 3 months ahead. And it's really the disconnect between disciplines because they don't know. It's lack of knowledge of what you need to, what information you need to provide for who. We talked about this inadequate analytics, and the other one is that quick and dirty phase one. I think it really happens a lot because particularly for smaller companies, you don't have money, you want to get there very quickly, but it really bites back if you don't know what risk to take, and if there aren't enough expertise to say, look, this one you really do not want to, this risk you do not want to take, and you have to watch for that. So anyway, with that, thank you for the attention. So questions?
Well, thank you very much for the presentation. It was very interesting, even if I didn't participate in many of those kind of crisis.
Yes.
So, talking about the research and development, communication, and I know what you are talking about because sometimes it feels that when a product jumps on the other side, so move from the basic research to the development, and it go basically jumping from one fence. Once you jump the fence, the communication is broken. But I think my question is more like, do you think for your experience, it's important that you get involved into the product development prior to the time that jump the fence?
Yes,
or
absolutely.
It's okay for you that once you jump the fence, it's in your hand and you focus on whatever, and eventually can only ask questions. But tell me, how does it work?
Sure. And so I didn't really kind of give you my background. So I've been in biotech industry for about now almost getting to be 30 years. I'm much older than what you think. But and I was at Genentech before for about 10 years. I was at Shire for about 10 years. So 2 big companies with lots of products. I was involved in the development of products from beginning to the end. And actually, one of the examples that I mentioned to you was there were some lessons learned from these examples where there was organizational change after that in terms of having a team that has on that team of basically preclinical, before it's a development program, that we started having people from development sitting on those teams before a decision is made for a molecule to come into development. Because it became very apparent that it's very inexpensive to do. Put one person who understands, a couple of people who understand on your team, do a few small studies. This stability study that I said, it took me a couple of weeks and a few HPLC runs. It wasn't a big deal, but if you don't know the discipline, it never happens. It may not progress things. So both companies actually, because in both of those companies that I've seen, and these are big successful companies, organizationally we actually changed how we did things, where development is involved before as part of the decision making to come from research into development. So we had to put together packages for senior management review that had an entire, like a CMC section actually in there. Is this a developer program? Do you have, what are the risks? How are you going to manage that? And so, just as you did clinical part and marketing part and whatnot. So, absolutely, it's important. Yeah.
And actually, I think it's very interesting because on the other end, we always think that the big companies, everything was perfectly fine. And they do make mistakes.
Yes.
And actually, their mistakes is even more expensive because they are millions of dollars.
Right. And it has major number impact. Yes.
And even if there is a lot of experience there,
yes,
of course they can hire.
Because they're silos. You mentioned silos, right?
Yes.
Again, there may be experience, but if they're not connected, if somebody doesn't ask the right question, you wouldn't know.
Yes.
So, interesting. Absolutely. Okay, thank you. I'll just put it here.
Do anybody have any question? I think we can do the raffle now. You are going to be